Okay, so this is the review sheet for cells and movement across the membrane. So what I'm going to do, I'm going to talk through it while writing through the answers. So top box then, it says draw and label a plant and animal cell. So I'm going to start off with the animal cell. So I'm going to draw a cell membrane. In the middle, I'm going to have a nucleus, and then I'm going to draw mitochondria. So if I'm going to label it, I've got my cell membrane, then I've got my nucleus, then I've got mitochondria, and I have cytoplasm. Okay, if we're going to draw a plant cell, then I'm going to draw it next to it, and then I draw my plant cell, cell membrane, then we have our nucleus, vacuole, chloroplasts. Sorry guys. And then we have our mitochondria. So we have the exact same labels. So we've got my nucleus, my cell membrane is my inside line, cytoplasm is the space that holds it all together, mitochondria provides our energy, and then we have extras. We have our cell wall, we have our vacuole in the centre, and then we have our chloroplasts. Okay, so there's our plant and animal cell. Then we've got how do we calculate magnification. So magnification on microscope, or click the word then, we would do the eyepiece lens and we would times it by objective lens. Okay, that's how we calculate magnification. If I'm going down, what do each of these words mean? So, if you start with cell, cell is our building blocks of all living things. And then we have tissues. They are a group of cells working together. Then we have an organ, which is a group of tissues working together. And then we've got organ system, which is a group of organs working together for a specific function. For a function. And it says, can we give an example of each of the words above? Okay, I'm going to do that in like a different colour, and I'm going to do it up the top. Okay, so an example of a cell, we could have sperm, we could have muscle, we could have liver cell, okay, any type of cell. Then we have tissues, so tissues, we could have muscle tissue, main example. Then organ, well, we can all name organ. We can go for the heart, we can have lung, brain, etc. Organ system, then I'm going to go for the respiratory system as an example. Okay, then we've now got the question, what is an enzyme? So an enzyme, one is a protein. It's made of proteins, but it's also a biological catalyst. And if we're not sure what a biological catalyst is, it's something that speeds up the rate of reaction. And what's special about it as well, that it's not used up in the reaction. So we can use it over and over again. Okay. Then we've got describe how the lock and key hypothesis works. So lock and key hypothesis is just like a lock and a key to your house, that one key is going to fit the lock. Okay, so if I was going to draw that, I would have, there's my enzyme. We know I'm not an artist. Okay, and then we have my substrate. From that then, we have them joined together. Where this would be called an enzyme substrate complex. And then from there, we'd have our enzyme. Getting dodgy, and we might have then our products. Okay, where essentially my substrate, I've drawn it that is split, so we now have products. Okay, and then we're going to say then if we had a circle substrate, so if I draw on it now, if I had say a circle, okay, he's not going to fit into my enzyme. He's a completely different shape. Okay, so lock and key hypothesis is that one type of substrate will fit and bind with one specific enzyme. Okay, one type of substrate can bind to one specific enzyme, like with a lock and a key. So how can enzymes be denatured? Okay, main one, we look at high temperatures. Okay, so at high temperatures, enzymes are denatured, and then let's look at what that means. Okay, so what that means is, I put denatured. Let's have a look at my spelling, because denatured. There we go. So what that means is the active site changes shape, and as it changes shape, the substrate cannot bind with the enzyme. Because the enzyme can't bind, no reaction can take place. So if we see it on a graph, we tend to see it as my graph, and we go up and then it goes back down. And it starts to denature from around 40 degrees, okay, the enzymes will become denatured. So it'll decrease, all the way up until no reaction can take place, because every single enzyme is denatured. Then we've got, what is a specialised cell? So it's, a cell adapted for a specific function. And then we're going to look at how as well. So sperm cell, it's got a tail. What is a tail for? It needs to be able to swim to find the egg. It also contains half the number of chromosomes, okay, which is needed because we get half from mum, half from dad. And we also have enzymes in the head, and the reason then is to fertilise the egg. Wrong one. to fertilise the egg. Then we've got an egg, that it contains a yolk, and it contains a yolk and it's the nutrients for a developing baby. It also then contains half the number of chromosomes, because we get half from mum and we get half from dad. Then we've got a root hair cell. This is where it confuses people. Root hair cell is a plant cell, okay? It's not an animal cell that's on your head, okay? So root hair cell. So what its job is, it needs to absorb water. So to do this, it's going to have a thin cell wall, so it can get as much water as possible. It's going to have no chloroplasts. And the reason that is then, does it need it? There's no sunlight underground, so there's more space then, so it can absorb water. And it's going to have a large surface area. And again, large surface area, because it wants to absorb water, and as much water as possible for the plant. Red blood cell then, so it's got no nucleus. Okay, it's got a large surface area, so it can hold more oxygen, and it contains hemoglobin, which helps bind to the oxygen in the cell. Okay. Now we've got what is diffusion. So diffusion is the movement of particles from high concentration to low concentration Okay, and then osmosis is the movement of water from high concentration to low concentration through a semi-permeable membrane. Then next question, what is required for osmosis? Required for osmosis, we need a concentration gradient. Otherwise we will get no net movement of water. Okay, we need to have two solutions with different amounts of water. And then what is active transport? It's the movement of particles from low concentration to high concentration against a concentration gradient. And then, how is it different from osmosis and diffusion? Well, active transport requires energy. And the reason that is because we're going against the concentration gradient. Think of it as we're going from low to high. You've got the bottom of a hill on your bike. You've got to pedal all the way up the hill. Okay, it's going to require a lot of energy. Whereas osmosis and diffusion, they are passive processes, so they don't need any energy. And the reason that is, if you use the analogy of a hill on a bike, if you're at the top of the hill from high and going down low, you can just start going and the wheels will go for you. Your legs can be out, you haven't got to pedal. Okay? Give an example of osmosis, diffusion and active transport. So diffusion would be of carbon dioxide and oxygen going into our lungs, into our cells. Osmosis would be water into roots. It would also be water into our cells. Okay, and then active transport would be like minerals into a plant cell. So like potassium into roots. Okay, so that should be our cells movement across membrane review sheet complete. Thank you.